Welcome back, students. So today we'll be doing the termination point. So the termination of DNA replication. All right. So as I have told you that the DNA, for example, this is a DNA. All right. The two strands of DNA will get separated and form a bubble-like structure like this, isn't it? The second step. All right. In second step, the helicase enzyme will come and form a bubble-like structure here. It will start melting the DNA strands, and then the DNA strands get separated. Right, like this. We have separated the hydrogen bonds A, T, C, G, and that's why the strands of the DNA has got separated, and this is how it will form the melting of DNA. Now coming to the termination point in E. coli. For example, we are taking the E. coli. There are specific sites in the genome where the replication forks and the termination sites will happen. All right. So, for example, if we are taking this particular example of E. coli, here we are taking the two replicating forks. This is one replicating fork, second replicating fork. All right, where the melting of the DNA has happened by the helicase enzyme DNAB. All right, and we have shown the two termination site. What is a termination site? A ter site, which is known as a termination sequence site, which spans around 24 base pairs. All right, for example, this is a ter A. All right, ter A, ter B. Okay. So this is the specific site where the termination happens. This melting of DNA will meet at this point, and then the termination happens. What happens? The two strands of DNA, they'll open themselves, meet at a point where the termination happens. That means the DNA replication stops. We have to block the process, otherwise it will keep on going the replication. The DNA strands will keep on moving, the two strands of DNA will keep on separating and make the genome. Like this, in cells we have to stop the process for the DNA replication. Okay, otherwise mutations will happen. Positive, negative mutations are there. Here the negative mutation can happen. So for everything we have to stop. Same wise, in replication process, the termination site is there where the replication terminates. All right, and this ter site is having the permissive end and the non-permissive end. For example, this site, this is a TUS protein, because a termination site cannot terminate the DNA replication alone itself, so it needs the another protein known as a TUS protein. TUS protein. All right, this TUS is a termination utilization site protein. All right, termination utilization site protein. So TER and TUS both will meet and make a structure which looks like this. This is a ter A. For example, we have just given a name A and B, and ter A has a permissive end TER A and TUS C, where the TUS C protein is having, this is a permissive end. A permissive end, and this clamp-like structure is a non-permissive end. Non-permissive end. Okay, permissive end which allows this replication fork to move further, while the non-permissive end will stop or block the replication bubble. That is, the separation of the two strands will stop here. This has the capability, this non-permissive site of the TUS protein has the capability to block or to stop the entry of this bubble-like structure, the replication fork. All right. So when this, for example, this replication fork will move in this direction, right, and this in this direction. This is a clockwise direction and anti-clockwise direction. Anti-clock. So when this anti-clock replication fork will move this side, what will happen? It will come and encounters the permissive end, right? This permissive end will allow the replication fork to move further and meet this another TER B, all right? While this replication fork, when it keeps on, the helicase enzyme will keeps on separating the bonds here. It is separating the bonds, okay, hydrogen bonds, the ATCG pairs. So when the helicase enzyme is entering this side, what happens? This non-permissive end, it will encounter the non-permissive end and it will stop the blockage. So what is it will stop the process. So what will happen? This replication fork is moving this side. This permissive end is allowing the replication fork to move further and meet this point, isn't it? After allowing, what does this TUS protein do? It will drop itself down. All right. It will permit the replication fork, and then when it comes here and this one here, what will happen? The helicase enzyme cannot pass through this, all right, and this non-permissive end will flip the nitrogenous bases here. For example, these are the nitrogenous bases present here. What this non-permissive end will do? It will flip the two nitrogenous bases upside. Then what will happen? The helicase enzyme cannot pass through this flip point. It needs nitrogenous bases. It needs nitrogenous bases to which it can break. But here, if it will not find any nitrogenous bases, because this non-permissive site has flipped the nitrogenous bases upside, so helicase enzyme doesn't have any base pair to break, right? So it will stop it here. It will stop the process here. All right. We can see it has flipped it here, and this non-permissive site has blocked the way of helicase enzyme. All right. This is how the termination happens of DNA replication. So no more process going on now. The two base pairs that are flipped like this, helicase enzyme cannot now pass through this, and the other replication fork has also come and stopped here. Okay, this is what is known as the termination site. Termination of the DNA replication. This is how the termination of replication meets here and terminate the process. All right. So now I have covered everything. That means the DNA the initiation, elongation and termination of DNA replication. So here I end my my class. So please subscribe my channel.